Good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So here we are discussing the last two divisions of our plant kingdom, that is Gymnosperms and Angiosperms, from which we can expect one or two questionnaire in our final NEET examination. So yes, let's begin with first Gymnosperms. Gymnosperms are well described as embryophytic, tracheophytic, archegoniate phanerogams. Okay. So embryophytic because they have embryo, tracheophytic because they have vascular bundles called tracheids, and archegoniate because their female reproductive structure is archegonia and they are flowering plants, so they are called as phanerogams, and these are said to be primitive. Just a second. So this will have primitive seed formation, wherein seeds are without seed coat. How? Because before fertilization, that is pre-fertilization, ovules are not enclosed by ovary walls. So here if we have to consider the ovules are not enclosed by the ovary walls. As such, we all know that ovule post fertilization will turn to what? Seeds. Seeds will not be covered by seed coat or fruit wall. Clear, right ma? Next, plants are medium sized or tall trees or shrubs. Habitat, if you see habitat of gymnosperms, they are terrestrial, mostly they are mesophytes. Smallest gymnosperm is Zamia, largest gymnosperm is giant redwood tree, that is Sequoia. Living fossil of gymnosperm is what? Ginkgo. Ginkgo is the living fossil in gymnosperms. When we have to look into the vegetative structures of gymnosperms, those are root, stem and leaf. Okay. Roots will have two types of associations, mycorrhizal roots and coralloid roots. Mycorrhizal roots are seen in pinus, okay, wherein the roots are associated. Both of them will have taproot system only. The roots will be associated with the mycorrhizal body. This mycorrhiza will increase the surface area, thereby which helps in the more absorption of water and minerals from the soil. So as the fungal association with the roots of pinus increases the surface area and increases the absorption of water and minerals, that is the benefit of this symbiotic association in the pinus. In Cycas, if you see, the symbiotic association is with cyanobacteria or BGA. Here it is with fungi and here it is with BGA or cyanobacteria. Okay, BGA or cyanobacteria. For example, Cycas. Remember these two examples, ma? Pinus, in Pinus gymnosperm, this is having fungal association, that is mycorrhiza. Cycas is having blue-green algae, which are coralloid roots. Both the root systems will not have root hairs or root caps. So there is an absence of root hairs and root caps. Root hair and root caps are absent in the root system of this. Next, stem, if you see, unbranched stem is seen in Cycas, branched stem is seen in Pinus and Cedrus. Okay, unbranched stem in Cycas, branched is seen in Pinus and Cedrus. Next, stem, in stem you can see U-stele, which is an advanced stele arrangement. Vascular bundles are conjoined, collateral and open, which aids in secondary growth, as vascular cambium is present in between xylem and phloem. In between xylem and phloem, vascular cambium is present, which aids in the secondary growth of roots and stems. So xylem, in xylem if you see of this vascular bundles, vessels are absent. Vessels are absent in the xylem. So what tissues are present, ma? Tracheids are present. Tracheids, xylem fibers and xylem parenchyma. Phloem, if you see, companion cells are absent. In the place of companion cells, albuminous cells will be present. Sieve tubes are absent, but then sieve cells are present. sieve cells are present, sieve tubes are absent, companion cells are not present, albuminous cells are present. So, this is about the vascular bundles which are observed in the gymnosperms. Secondary growth is possible because of the vascular cambium or open type of vascular bundle. Next, leaves, if you see, leaves are pinnately compound leaves are present, for example cycas, whereas needle-like leaves are seen in conifers. This needle-like leaves will reduce the surface area, okay. Needle-like leaves will reduce the surface area, which is the best adaptation for xerophytes. Next is fan-shaped leaves will be observed in ginkgo. In ginkgo, fan-shaped leaves are seen. So, leaves are adapted to withstand extreme temperature, humidity and wind. Thick cuticle and sunken stomata are observed, which is the best adaptation to reduce what? Water loss or water stress. And then, now let us come to the sporophytic structures. So, the first heterosporous condition is observed in pteridophytes, and then heterosporous condition is also observed in gymnosperms. So, yes, once let us recall the evolutionary perspective with this homosporous and heterosporous condition. Bryophytes are completely homosporous. Pteridophytes, few of them will show heterosporous condition like Marsilia, Azola, Selaginella and Salvinia. Whereas, if you see gymnosperms, completely heterosporous condition only is observed. For example, microspores and megaspores. These spores are present on the leaf structures in the form of cones, like how you see in the equisetum of pteridophytes. Same cones or strobili are present, but they are on those leaf structures, so they are called as what? Sporophylls, which are arranged spirally on the axis, called as strobili or cones. Sporophylls, we have two types based on the type of spore it is carrying. Okay, we have microsporophylls and megasporophylls. Microsporophylls, if you have to look into, they are compactly arranged to form what? Male strobili. Male strobili is also called as microsporangiate or male cone. Okay, this is also called as microsporangiate or male cone. Microsporangiate is having microspore mother cell which is diploid. Okay, microspore mother cell is diploid. This undergoes sporic meiosis and forms 4 microspores which are haploid. After sporic meiosis, 4 haploid microspores will be formed. This develops into what? Male gametophyte, that is pollen grains. Okay, so microsporophylls which are compactly arranged, also called as what? Male cones, which are also termed as microsporangiate or male strobili. They will be having diploid microspore mother cells which undergo sporic meiosis and releases microspores which are haploid. They develop into the male gametophyte, those are haploid mature pollen grains. Right, ma? Next, megasporophylls. Megasporophyll is said to be what? Female strobili, also called as megasporangiate or seed cone. So, this is also called as female strobili, megasporangiate or seed cone. So, here if you see archegonia, inside archegonia the cells which are present are called as nucellus. Nucellus is diploid. Okay, nucellus cells which are present inside the archegonia are diploid. So, here you have 2 to 3 multiple archegoniate structures. Once again you listen, ma, this is the complete female strobili or female cone or seed cone. Inside that you will be having 2 to 3 archegonia, multiple archegonia are present, and the cells which are present inside this female cone are what? Nucellus cells. These nucellus cells are what? Diploid. Nucellus cells, one of them will be differentiated into megaspore mother cell which is diploid. Okay, if nucellus cells are diploid, one of it is differentiating into megaspore mother cell, that will also be diploid, and this will undergo sporic meiosis, forming out to be 4 haploid megaspores. Okay, 4 haploid megaspores will be formed, of which 1 megaspore is functional and rest 3 will be degenerated. This functional megaspore Okay, functional megaspore will develop into, functional megaspore is what? Haploid, right ma? So this functional megaspore will develop into multicellular female gametophyte, which will bear again 2 to 3 archegonia, which is retained within the megasporangium. So as this haploid cells or haploid haploid female gametophyte is retained within the megasporangium, endosperm here is formed pre-fertilization, unlike angiosperms in which endosperm is formed post-fertilization. Triploid endosperm in angiosperms is formed post-fertilization, but in gymnosperms, endosperm is formed pre-fertilization, okay, before the fertilization. Because of this, it is what? Haploid endosperm, which is nutritive. Clear, right? So megaspore will give rise to endosperm. Megaspore, which is haploid, that gives rise to endosperm pre-fertilization. Now, if you have to look into the pollination, many modes of pollination will be there, and type of oogamy is siphonogamous oogamy. Till pteridophytes, we have observed zooidogamous oogamy, but here in gymnosperms you can see siphonogamous oogamy, wherein zygote will differentiate into embryo, ovules will be formed seeds, but those are naked because of the absence of seed coat, because there is no ovary wall before fertilization also. Now in cycus, let us look into few characteristic features of gymnosperms which we can see in pteridophytes as well. So here if you see ma, cycus will show both siphonogamous oogamy and zooidogamous oogamy also, important for EAP set students. Male gametes here are in cycus multiciliate and they are carried by what? Pollen tube. Form characters include sarcinate venation of young leaves. Okay, in gymnosperm sarcinate venation is seen, presence of ramenta, presence of archegonia. Gymnosperms are divided into 3 classes. So these 3 are the common characteristics which coincide or resemble with the pteridophytes. Here if you see the gymnosperms, it is having 3 classes of the gymnosperms. For example, Cycadopsida, Coniferopsida and Neetopsida. Cycadopsida, Cycus is the example, Coniferopsida, Pinus, Neetopsida, Neetum. Clear, right ma? Next. Here once you recall the life cycle completely, the mature plant body is what? Sporophytic plant body, and in gymnosperms you can see what type of life cycle ma? Diplontic life cycle. Diplontic life cycle. So here diplontic life cycle you can see pollen cone and ovulate cone, both of them are diploid, okay. And then here you can see microsporangium which undergoes sporic meiosis, and this forms haploid microspores which differentiate into haploid pollen grain and then to haploid male gamete, which show wing type of pollen grain in Pinus. Pinus will show winged type of pollen grain, which is an important question, okay, once asked in the final examination. Next, ovulate cone or seed cone which is diploid. This megaspore mother cell also undergoes, megaspore mother cell which is diploid will also undergo what? Meiosis, the sporic meiosis, and they form what? Haploid megaspores which differentiate into the female gametophyte. Post fertilization they form what? Zygote will differentiate into an embryo and forms a seed. This seed will be naked without the seed coat, okay. So this is all about the gymnosperms. Now let us look into the angiosperms. Angiosperms, whatever we are reading over here, we will learn again in sexual reproduction of flowering plants of NCERT 12, okay. So this is just like a recap, okay. Now angiosperms, how you can describe it in a better way? They are embryophytic, non-archegoniate, because here the female sex organ is what? Gynoecium, not archegonia. They are vascular, they are fruit bearing and seed bearing as well, and they are phanerogams, spermatophytes. Gymnosperms also we can call as spermatophytes. So smallest angiosperm will be what? Wolffia. Smallest gymnosperm will be what? Zamia. Largest angiosperm is eucalyptus, largest gymnosperm will be sequoia, Okay. So, this is about angiosperms. Vascular bundles include xylem and phloem. Xylem has xylem fibers, tracheids, vessels and xylem parenchyma, whereas gymnosperms does not have vessels. In phloem also we have 4 different types of tissues ma. Phloem fibers, sieve tubes, companion cells and phloem parenchyma, whereas in gymnosperms instead of companion cells we have albuminous cells. The reproductive organ of angiosperm is what? Flower. Flower will be having 2 essential whorls and 2 non-essential whorls. Okay, they have 2 essential whorls and 2 non-essential whorls. Essential whorls are, they have 2 essential whorls and 2 non-essential whorls. Fine. Essential whorls include andrecium and gynoecium, male and female reproductive structures, andrecium and gynoecium. Here you can see the andrecium clearly. Okay. The male reproductive structure, that is antheridium, which has anther, bilobed anther and filament. Female reproductive structure, that is gynoecium, it will be having stigma, style and ovary. Inside ovary, ovules will be arranged. Non-essential whorls include sepals and petals. Okay. So, the clear description of sepals, petals, arrangement of ovules inside the ovary, about male and female reproductive structures, clear morphology we will be learning in morphology of flowering plants. Okay. So, this will be the connecting concepts, connecting concepts of morphology, this plant kingdom angiosperms and sexual reproduction of flowering plants of NCERT 12. Always you have to connect the dots between chapters and learn. And here if you see male gametophyte, that is pollen grain landing onto the stigmatic surface, compatible stigmatic surface will be giving out or generating out the pollen tube. At this time only pollen grain will undergo mitotic division, releasing out what? 2 male gametes. This male gametes will reach the ovule. Inside ovule, embryo sac will be present. Inside embryo sac, this male gamete will be fusing with the zygote. 2 male gametes are there right now. One will fuse with zygote and one will be fusing with what? Secondary nucleus. So, here if you see, this is the structure of what? Embryo sac. This is the micropylar end. Over the micropylar end you have egg apparatus, single egg cell and 2 synergids, and at the chaliceal end you have 3 antipodals. At the center you have polar nuclei or the central cell. Double fertilization, it is called as double fertilization because 2 fusions happen. One is called as syngamy. Syngamy is the fusion between the male gamete and the egg cell, which forms a diploid zygote that differentiates to a diploid embryo. Whereas triple fusion is one male gamete fusing with 2 polar nuclei. As 3 nuclei are involved over here, this is called as what ma? Triple fusion. Triple fusion will result in primary endospermic nucleus, which is differentiated into an endosperm. Clear, right? Next, life cycle of an angiosperm. Angiosperm life cycle will be strictly sporophytic, and the mature plant body, the main plant body is sporophyte only. In both gymnosperms and angiosperms, sporophyte is independent and gametophyte is dependent on the sporophyte. Hardly haploid gametophyte stages you will see in haploid microspore and haploid megaspore, and here haploid pollen grain and haploid egg cell. Those only will be the haploid stages in both gymnosperms and angiosperms. That is why the life cycle is diplontic life cycle. So, here what is the life cycle ma? Diplontic life cycle. Just a second. So, here it is showing diplontic life cycle. So, here the diplontic life cycle, if you see, the microspore mother cell forms microspore tetrad. This forms a tube cell and a generative cell. Tube cell is nothing but the vegetative cell. This generative cell again undergoes what? Division, forming 2 male gametes of a pollen tube. Okay, this landing onto the suitable substrata will give out 2 male gametes. Till here it is clear, right? Now, inside the ovary, ovules will be present. Inside the ovule, embryo sac will be present. This embryo sac, 2 male gametes are there right now. One is fusing with what? Egg cell, and one is fusing with the polar nuclei. This results in the formation of zygote and endosperm, which forms endospermic seeds. Okay, zygote will develop and mature. Zygote will develop and mature into an embryo, and endosperm will be maturing into cellular endosperm, which finally forms a seed. Seed on germination will develop into a young sporophyte again. So, this will be the life cycle of angiosperms. Now, we have 2 classes of angiosperm division. Those are dicots and monocots. The clear morphological differences between them are very simple. Dicots will be having 2 cotyledons, monocots will be having only 1 cotyledon. Dicots will have branched veins, monocotyledons will have parallel venation. Dicots will have 4 or 5 petals, monocots will have multiples of 3 whorls. So, lastly, about alternation of generations in plant kingdom. So, in plant kingdom completely, if you see from algae to angiosperms, especially in angiosperms, we have alternation of generations where gametophytic generation will be alternating with sporophytic generation. So, here diploid sporophyte generation starts from zygote to the mature sporangium. Sporic meiosis will start the haploid spore, which begins the haploid gametophytic phase, and these 2 phases will be continuing, which results in what? Alternations of generations. Okay, so this is all about the plant kingdom. The life cycles I will be discussing in shorts, wherein we can discuss the exceptions as well. So, that is all for today. Tomorrow we will be starting morphology in flowering plants. So, stay tuned and thank you.